The title of this presentation is Transcranial Photobiomodulation Dosimetry, Integrating Human Data and Penetration Model. I'm Paolo Cassano. I work at Massachusetts General Hospital in Boston. So here are my disclosures. Of note, I'm a co-founder of a company in the light photobiomodulation field. However, I'm not employed by this company. So first of all, a brief introduction about photobiomodulation. It's a technique to stimulate and neuromodulate the brain through near-infrared light. Now here you can see the different type of lights. Now near-infrared light is depicted in red, although it's invisible. The point of this figure is that near-infrared light penetrates deeply into the cell, into the tissues, and has its own photoacceptors at the level of the mitochondria. One of the most important byproducts is the stimulation of the respiratory chain, which leads to increased production of ATP. However, there is a transient increase of respiratory free radical and also transcription factors like the NF-κ beta. Those have effects on activation of other transcription factors and therefore a cascade of overall not only high energy secondary effects, but also antioxidant, anti-apoptotic, anti-inflammatory effects that are very important for the health of the cells and also for the neurons. They're also specifically synaptogenetic and neurogenetic effects. So neurogenesis is stimulated, for instance, through BDNF. This is a close-up look at the membrane of the mitochondria, looking at the respiratory chain. And here is the photoacceptor, the primary photoacceptor for near-infrared light. And near-infrared light is that light of the infrared spectrum that is particularly close to the visible. And over here is a higher resolution of the cytochrome C oxidase. The effect of the light is to basically transfer energy, light energy, into electron transport and lead to a polarization of the membrane, which then leads to the ATP formation, as there is a proton transfer involved. ATP is a high energy molecule that is eventually utilized by the cells. Now, this is what we do. This is what we are interested in. However, why did we want to do this study about simulations, computerized simulation, computerized modeling of the penetration of the light? Well, the issue is right here. In front of us, there was this gigantic, colossal study through 3 phases, the NEST 1, 2, 3, that look at the effect of photobiomodulation for stroke. A lot of excitement, a lot of investment, up to 40, 50 million were invested in the company that was leading this study. However, at the end of the day, it led to futility. And there was a lot of hope. And the animal models were very promising because they showed that there was an option to re-energize the brain right at the time when ischemia was happening and when the blood flow wasn't quite bringing the right energy, the right oxygenation. So why didn't it work? In this important article, Dr. Lapchak and Boitano made an important statement that animal models are not or cannot be directly translated into the human situation, because oftentimes the head of the humans and the skull are different and the thickness matter. So both our group at Mass General Hospital and the group of Chen Feng at Northeastern University were dealing with this dilemma of what to do. And we wonder if computerized simulation models could provide us with more concrete, tangible information to use for clinical studies to inform the dose and not to come back to the same pitfalls of the previous studies, which are very disappointing and also risk altogether to really sink the entire field of transcranial photobiomodulation. So the group of Chen Feng had a simulation model. But before we get there, I think it's critical here to say that there are already very precise data from cortical neuron cultures of what dose is needed to make a difference. And these are studies of the group of Luis de Taboada, where they showed here on the axis the fluence, in which is an energy density measure at the cellular level. And you can see here on the y-axis the ATP production. And this was a fluorescence mechanism. And you clearly see that as the dose increases, there is an improvement in the total ATP level. So a clear marker of the effect of the light at the neuronal level. And then after that, a decrease in the overall effect. And you can see that 3 joule per square centimeter seems quite a right dose to aim for. Then when you look here at the mitochondrial membrane potential, you can see that the effect can be seen as a significant effect as close as 0.3 joule per square centimeter at the cellular level, and it's even more obvious at 3 joules. So we have clear data of what we should be aiming to in terms of energy deposition, energy density. However, how do we get there? And that was the question we were wrestling with the group at Northeastern. So they had a Monte Carlo model, a mesh model of the head based on computerized simulations. It's MCX because they found a particular algorithm to speed up the entire system and really provide information in real time in a fast manner. So first of all, we chose some target brain regions. Here's in green the dorsolateral prefrontal cortex, and in red the ventromedial prefrontal cortex. This purple area is the frontal pole. There's different ways to define the ventromedial prefrontal cortex, including that area, the purple, or without. Here, another picture to simulate, show here the location of the device and the light sources in our simulation over F4, F3, and over FP1, FP2. These are EEG points. So here, another way of modeling. We want to really to be close to the clinical situation. And here's, if you put the device on basically F3, F4, you have the device higher up, and or if you put it closer to the frontal pole, aiming at the ventromedial prefrontal cortex. Of note, the distance from the source to the target is much greater when you use the frontal pole as a site, as opposed to when you use higher sites on the forehead close to the hairline. So what we wanted to do is make these predictions and then go back to our data and see if the clinical data could be a reasonable testing ground for our predictions, and if the clinical data supported the predictions that we had made with computerized modeling. If that's the case, then you could imagine later on using the computerized modeling in a prospective way, and therefore inform, as I mentioned before, the dosimetry for future studies, and maybe even the clinical applications, although photobiomodulation is not an approved treatment for psychiatric condition. The good news is that our group at MGH, also in collaboration with New York University and with the group of Dana Iosifescu, had an extensive group of series, I should say, of studies. And all of these studies had fairly similar parameters. The wavelengths around 830, mostly continuous wave, irradiance in between 33 and 55 milliwatt per square centimeter, fluence in between 60 and 65 joule per square centimeter, duration of treatment ranging from 20 to 30 minutes, window of treatment was obviously a little broader when we were using certain devices, so there was certain variability, but not huge, and total energy was also in the same ballpark, 3 to 2 kilojoules per session. Now, the first simulation was about varying the dose. So this is a busy table here, but if you follow this table vertically, you can see on this side that you're looking on the parentheses, the total energy delivered, and that's an easy ballpark number. So you go from 2.5 kilojoule, 1.5, 1 kilojoule. Now, if you look at the effect on the median energy deposition, it goes from 0.5 to 0.3 to 0.2. So clearly, you get half of the energy if you go half of the energy on the skin. So this is on the skin, the total energy, and this is the predicted energy density median at the neuronal level. So clearly, and obviously, a dose effect when you were decreasing the dose on skin. Interestingly, this is the median related to the upper quartile of the voxel, of the cortex that you are aiming to. So it's telling that the values might be and are expected, in fact, to be much lower in the lower quartiles. So then we went to the data, and we had a healthy subject group of 10 subjects where we looked at 2 different doses. All parameters were the same. The patients were the same. We were tested with 2 different doses. And what we found is that, interestingly, at a certain dose, we could have a significant potentiation of the EEG activity in these healthy subjects, especially in the gamma range, right after the photobiomodulation, here expressed with the red curve. This is on the Y is the power density of the EEG signal, and this is before the light. However, when we cut the dose by 1/3, this effect was lost. And so here we clearly saw a confirmation of the prediction we had made in the computerized modeling. Another prediction that we made through the computerized modeling is related to the effect of age. So because of the development of the head, the change of the size of the head, of course, from the child to the adult, this is the age in the x-axis. There is an increase in the thickness of the tissue. So, and the y is the thickness measured in millimeters. So you can see that that thickness of tissues in between the surface of your skin and the surface of the brain, it progressively increases over time, even as adults age and up to the elderly. That's obviously an issue because there is what we call an attenuation of the light signal that is related to this change. And now, if you think about it, you could have a group of healthy subjects around their 20s, the typical volunteers that come in the healthy subject studies. And then you can have the fairly overall typical middle-age sample around 45 to 50. That is the typical sample that you encounter in a clinical trial. And the thickness is not the same. And therefore, there might be changes in penetration. Over here, you can see, in fact, modeling now on the X, the thickness, and on the Y, the average energy density deposition. Now, if you go back, if you see this dotted line, I put here what's the expected thickness of the extracerebral tissue. So around 45 to 50 age, so year old, you can expect roughly 12.5 millimeter on average, but when you are in your 20s, you can expect probably around 8 or so millimeter on average. Now, if you translate it over here in this other plot, now the people who are in the 20s are here, the people who are in 45 and 50 are here. So now go to the red and the green line. This is basically on a logarithmic scale, the progressive attenuation of the light as the thickness increase. For this example, we looked at the target from F3, F4 on the dorsolateral prefrontal cortex. So F3, F4 are the points, the EEG point on the skin, and the target area is the dorsolateral prefrontal cortex. And you can see that going from your 20s thickness of the extracerebral tissue to your 50s actually is roughly half a point on a logarithmic scale of attenuation of the light. So we expect a significant effect here related to age. Now we tested that looking at the same device, so identical setting and parameters, and we looked at the same group of healthy subjects we had discussed before, and a group of 49 subjects with a major depressive disorder. And we looked at what type of effects we could find here with the same type of treatment. And here we go again, the effect we just showed you before in healthy subjects, the potentiation of the electrical activity with only one session, immediate effect. same treatment repeated over time over 12 weeks. Now here in the X, you see the visit numbers. These were 12 weeks worth of visit. Over here, you have the total score of the QIDSI, depression score. And here you have 3 groups. And these 3 groups are patients who are randomized either to near-infrared light, sham and near-infrared light, or sham and sham. And you can see that basically, regardless of the repeated treatment that they were getting twice a week, these patients had no difference whatsoever, whether they were getting sham all the time or they were getting near-infrared all the time. These were basically mixing and interweaving. So at this point, our prediction was somewhat confirmed by this data that if you change age group from these healthy subjects to these MDD subjects, and besides obviously the condition from healthy to MDD, you find that you basically lose the effect, probably because of that extra thickness and decreased penetration of the light. Finally, we looked at another element here, which is the probe site. What if we change from the site on the dorsolateral prefrontal cortex that I'm pointing here to the site aiming at the ventromedial prefrontal cortex? Here in the graph, it's the red and green line show the attenuation of the light with greater thickness of the skull when the light is shed on a 3F4 for the dorsolateral prefrontal cortex. And here in blue, the most conservative evaluation of the light that is arriving onto the ventromedial prefrontal cortex when shed at the level of FP1, FP2, FPC. And you can clearly see that for that age group of MDD patients in their 50s who had a predicted thickness of 12 millimeter, if you go from one side to the other, you basically lose 1 logarithm of light. So dramatic effect. If you see here, we first of all discussed the impact of decreasing the total amount of energy. Remember, from 2.5 kilojoule to 1.5 kilojoule to 1 kilojoule per session, which obviously corresponds to the different energy density at the level of the skin, but also progressively lower energy deposition. Here are the median, 0.5, 0.3, 0.2. Now, think about the added effect of not only decreasing the amount of total energy you're putting on the skin, but also shifting your target, not just vertically, but also horizontally. So you're going from 0.5 to 0.3 in terms of predicted energy deposition if you decrease the amount of energy you're shedding, but then you're going from 0.3 to 0.07 if you're switching to the ventromedial prefrontal cortex. So dramatic effects when you combine the change in position of the probe and the change in dose. So we went to test that in basically 2 samples of depressed patients, similar age. And this was our first study. that was conducted aiming at the dorsolateral prefrontal cortex, related to, was a significant effect of the light here at the active treatment in red as compared to sham over 8 weeks. And here again, what I just showed you before, the effect of shedding the light on the frontal poles. Some light was also shed on F3, F4, but unfortunately was on hair, when the hair were also shielding the light. Moreover, the light was distributed among these two sites, so the light that was shed on the F3, F4 was obviously less because there was some split in between the two sides. So clearly, going lower the dose and also changing the position to the frontal pole was not a good idea, and our model was tested and confirmed by our clinical data. Now, so in conclusion, using computerized modeling with Monte Carlo simulation extreme is promising. It's promising, as I mentioned before, to predict adequate dosing in research studies to avoid, hopefully, those catastrophic failures that we've seen in the stroke literature, and can be useful potentially also whenever we use this treatment, the transcranial photobiomodulation, as an off-label treatment. One note that I like to make is, as you've seen, sometimes the median energy in the upper quartile of the voxel at the level of the cortex of the target was 0.3, 0.2, was really kind of bordering that fork that we've seen as most likely the active energy, the active range of energy deposition to have an effect at the level of cortical neurons. So this is still a question that is open in the field. Is this energy that is kind of borderline sufficient to produce an effect? And it seems it might. However, is the effect indeed related to direct engagement of the neurons? This is still a question that we need to answer. So finally, I'd like to thank all my colleagues from the depression program, as well as the crew of Dr. Feng at Northeastern University, the Nathan Klein Institute in New York, as well as the MGH Division of Neuropsychiatry. Thank you.